Moderating the panel, we have Anastasia from Brandwood. She also was the former team lead, branch president of Innovation Forum. So I'll pass on the mic to her. Actually, yeah.
Thank you. Thanks for inviting. So my name is Anastasia, and I'm business development lead for life sciences at Brandwood SciTech. We are a network of innovative science parks. So one of them is this one, Bright Building, but also we have presence in Manchester, like City Labs. We have presence in Leeds, Liverpool, Alderley Park in Cheshire, you might know, Birmingham, Cambridge, and we are expanding now to Scotland. So we're quite a big network. My role is to strengthen and expand the ecosystem, but I'm a translational medic by background. I worked in AstraZeneca, and then I specialized in cell and gene therapy. So if to be very honest, genomics and AI is not something I directly worked in, but I was really closely connected to it. Today we are joined by a great panel, having broad background, but directly or not in who work in genomics and AI. So I'm joined by Simon, Markella, Augustine, and Catherine. I will let you introduce, guys.
Me first. So my name's Simon Pearce. I work at Cancer Research UK Manchester Institute, and I'm a mathematician, then a mathematical biologist, then now a bioinformatician / data scientist, essentially.
I think the question somebody asks who will win would be really applicable for you.
I'm Markella Mikkelsen. I'm founder and CEO of Molmart. We are a diagnostic genomics company, so we actually bring all this amazing data to the patient. I started as a molecular geneticist with some of the more traditional techniques that were mentioned today, like microarrays and PCR. And I got into genomics in 2015. So when I realized what genomics could do for the patient, it was just an eye-opener for me. I realized how much more we could do than with traditional techniques. So that's when Molmart was founded.
Hi, I'm Agustin Guardiola. I work at Apis Assay Technologies in City Labs next to the MRI. I'm a physicist by training, and I work in the computational department at Apis. So looking at the computational methods, AI, and statistics that we use.
Hi, I'm Kath Felton. I'm a PhD student at Cancer Research UK. My project is in pancreatic cancer, and I'm mostly taking actually a proteomics approach. But I'm mostly here because I started out as a biologist and I'm making that transition into data science. And so I wanted to bring kind of that sort of feeling of how do we integrate new talent from different backgrounds into data science.
Thanks, everyone. Thanks for the introduction. So my first question is to everyone, actually. We heard great stories from Danuta and from the representatives of Pankhurst Institute, how amazing, and of course, Illumina, how amazing is science and how far we are now compared to where we were. So when it comes to where we are now in relation to drug discovery and diagnostics, how far did we go, where we are now? And when it comes to data, do people really use data, or is it sort of a hype word so that people can apply for grants or show that they collaborate? Or yeah, shall we start from that end now?
Okay. So from a student perspective, I think data is kind of thrown around but not really used. I think there's so many data sets in repositories and potentially even data sitting in your own lab that hasn't been utilized and analyzed properly. And so there's a lot of scope still left to happen for PhDs to start out as data projects and to kind of define where they want to go with their research from the data that's already available, rather than just diving straight into the lab. So I think that's kind of where we're at from a student point of view.
I would say that there's certainly both sides of the coin. So as we saw in the earlier talks, there is great advances being done with big data and machine learning. But as you just said, it's true there's huge biobanks of data. A lot of the time that data for widespread access is heavily pre-processed, which limits how much of an analysis you can do or how much of deep learning you can do on it. So there's certainly scope to do a lot more.
So from a diagnostics perspective, again, we have to be a little selective of the data. Of course, there's a lot of data out there, but we need to be very careful of what we choose and how we apply it, because everything we do has a clinical implication for the patient. So a lot of the new omics that we heard about today is probably a little bit too distant for us at the moment to be able to use and apply in what we do.
I definitely echo Kath's comment about data sets are generated like in academia often for like one specific project by one PhD student and analyzed a little bit and then never looked at again far too often. But the challenge is like combining data from multiple sources, and it just gets put in repositories that you can't then access. It's really hard to actually get base genomic data because of some of it for very good reason, because of the patient confidentiality kind of questions that doesn't then lend itself to being analyzed in big ways unless you've got a big platform that like Illumina have to help that.
Sorry, the UK biobank data is actually being reanalyzed on Dragon because the tools are better. So some of that holds back mining the data that you have.
Yeah.
So actually, on that question, I think to representatives to yourself from representative from the Research Institute and yourself, Augustin, as a representative from Scale Up or SME, where are we heading to? Does it mean that in, say, 30 years time, we will reanalyze again because we'll have Dragon, 2040? Or where we head, or are we going to dig out this data that hasn't been analyzed and it was piled on a bookshelf and now it's time to kind of dig it out and kind of use it? Where are we heading to then?
Yes, some of that will be complicated by different library prep methods or sequencing depth. As it gets so much cheaper to sequence, you can sequence more. So like coverage that was acceptable before is like no longer going to be cut it in terms of depth, for instance.
Yeah, I think a lot of this will depend on how the genomics technology advances. And for example, if we move from bulk sequencing to single cell sequencing, obviously we need new data sets for this, but ideally, yes, we'd have all of these omics data integrated together alongside with an electronic health record. And through AI, we'd be able to make informed clinical decisions that hopefully have a justification that can be interpreted by a person. It's not just a black box system that no one understands why this is the outcome.
Mm-hmm. And then when it comes to translation, say in APIS assay, how long does it usually take, or is there such a process at all, like of translation, right? We know when we do some kind of R&D and then we want to translate into commercialization, process. So, or we created a new diagnostic tool. How long does it take? And also, say, for instance, from Research Institute, is there a scope for it, or we just do research to improve patients' life, but not to create, say, more private entities?
So I would say that the process of getting from a proof of concept biomarker into a product is something we've personally found challenging. It's that you've got the reproducibility conundrum a lot of the time, where you change the instrument, you change the technique, or you simply change technology and suddenly the same samples give you different data and they're not necessarily reproducible. In terms of length of time, probably around 3-4 years, especially considering that we work in a regulated industry with the IVD regulations. It's a very lengthy process to validate and do all this.
Any comments?
Yeah, I think from maybe a research perspective, especially as like an early career researcher, when you're you can be the most entrepreneurial minded scientist in the world, but if you're kind of in the weeds of basic research, especially in biology, it can often seem difficult to think about the commercial application. And I think like for myself, kind of attending talks about incubators and thinking about kind of that idea of innovation into commercialization early on, it kind of gets your eye out and looking for things that might turn into something more commercial down the line, which is, I think, initially hard to find basically in basic biology research. But ultimately you kind of need that basic research to be the foundational tools for the translational work.
Marcella, and then as a representative of small SME, what are the challenges that small young companies, when they found this great R&D stuff that works, you commercialized, fantastic. What are the challenges and where academia could help? And maybe to Catherine then, what are the challenges in academia where industry could help?
So like any startup knows, cash flow is our main challenge. And especially in the area of genomics, where we have to have a very active R&D arm, cash flow without kind of serious investment does become an issue. I mean, it's fair to say that we wouldn't be here if we had not been preceded by academic research. So what we do is absolutely the direct result of what has preceded us. And one of the challenges that we face in this area is how we integrate all the new academic research in what we do, but in a safe and ethical way, because again, we are patient-facing, customer-facing. So that's kind of from the perspective of a commercial company.
Yeah, I think from an academic perspective, having expertise in industry and in entrepreneurship coming along early, I think would be helpful, or being able to start those dialogues so that, there doesn't even need to be an idea to have those conversations and to have those contacts, and getting the right people in the right room would be really helpful, I think.
That's where NAS can help you with the imagining. No, that sounds good. Simon, anything for you where Marcella could help you with?
I mean, the same issue of funding, of cash flow, is exactly a problem in research as well as in the small SMEs. But yeah, I don't know. I've not really dealt with companies like in. I know we do within Cancer Research UK, like the Cancer Biomarker Centre that I'm part of, but I haven't really been directly involved in those discussions or anything.
Okay, and then we'll go back to the question. I think Marcella mentioned about, you mentioned ethical questions. So the question is, who owns the data, and then how to get access to samples then?
That's a tricky one. We're still formulating guidelines. So in terms of what international guidelines are there, there's the American College of Medical Geneticists. So, but there are forums, out there that are still formulating guidelines of how we use data and what we can report and what we can't report. So where other areas are very kind of heavily directed by guidelines that have been, so for example, what you can report by microarrays or what you can report by cytogenetics, kind of very strictly governed by guidelines. This area is still a little bit open, and that is one of the challenges that we have as a diagnostics company. And to be honest, it's something that I've had in my kind of previous career in molecular work of where do you stop at reporting something that can potentially be actionable? And that's the key word for us, you know, what is actionable? So a lot of the information that we talked about today in the different omics, this is great. And absolutely, we want to, you know, keep an eye on what is happening, but at the moment, it is not actionable by a clinician. It's not something that a clinical geneticist or a counselor could sit with a patient and say, you know, you've got this, or you have a high risk of this. So that's kind of, I would say, our main ethical challenge. We talked a little bit earlier about reanalyzing data. That's going to be another challenge for us, because if something then becomes actionable, so say we issue a clinical report today and something becomes actionable in 5 years, what do we do?
So that means you have to reanalyze again, and then maybe
Yeah,
to
what if
question the
something that
report.
is, yeah, clinically very impactful for that patient? What do we do? How do we report it? How yeah.
I mean, and then once their data is in the public domain, at what point do that data not belonging to them, even though it's still their health data? I think those are questions that are really big, that don't have clear answers, and that will need some regulation and government oversight, but oversight into that kind of data could potentially lead to oversight into data in other industries that are less keen to have oversight to their data.
But that can create completely kind of different outcome, isn't it? Because say if patients have passed away, or for instance, they don't want to be involved in the study anymore for whatever reason, and vice versa, there is a next sort of, let's say, generation of potential volunteers who would like to be involved, we can get completely different results. Or
if the patient has passed away, it's not a problem.
oh, okay.
Fortunately,
Sorry to be
At
so
least one
cynical.
benefit. Okay.
So can I just, so we've mentioned microarrays a couple of times. Like, it's only 10 years or so that we've been really sequencing, like not just using, I would like my first postdoc position, I was doing microarrays, like, and now it would just be RNA sequencing without any.
I did single cell RNA sequencing in my undergrad project, so.
That was my last bench job, the last time I actually kind of touched a pipetted and touched the bench.
It's actually a really interesting comment that you say, because when I was actually on my placement in AstraZeneca, I was doing some of the just basic first research. Was like the first step was Western blot, and then my and then my manager PI came at the time and said, "Oh, Anastasia, you know, it takes you half a day to do it. I did my research for 4 years on that." I was like, oh my God, because I was thinking 4 hours or 5 hours on a high pace. It's a long time. And then I thought, you know what, it took him 4 years. So I think it's fantastic. I bet CRISPR will be like in a couple of years time, like for undergrads first year project, you know, something like that.
I gotta tell you though, microarrays are not dead. These things are like cats. They've got 9 lives. So we don't do gene expression on microarrays much anymore because RNA-seq is just better data, right? But pharmacogenomics, copy number, they are all incredibly cost effective on microarrays. And we're seeing, when we expected maybe 5-10 years ago as a company to be winding up microarrays, it's actually one of our biggest growth areas. Agribusiness, which isn't part of today's forum, but that's huge. It's really taking off. Genotyping livestock and plants is absolutely massive. So microarrays are definitely not dead and gone. I just banged on about sequencing today just because of the subject of the
Sorry,
forum.
can I actually ask, does it mean that in the future we can say you want this type of banana or this type of tomato, and then they will make it based on the sequencing?
Yeah,
Ooh.
that's a qualified yes.
That would be an expensive shopping then.
Yeah, probably.
Okay.
Just before we move on from that, Alex, I got a question. When you have, all these algorithms, that are performing these machine learning, so on and so forth on all these data sets, if say eventually it does get to the public health and it's used in the public health domain, if there is an error, whose fault would it be?
Who owns it?
Oh, well, I'm just saying, where's accountability? Is accountability because
Yeah.
it's an algorithm that is potentially.
Well, if there's IVD directives in place, manufacturer, isn't it? But bearing in mind Brexit, we don't have IVD in the UK right now. That's where the challenge lies. And this is why it's so important. It's really hard to build an IVD test for something like rare diseases because they are, well, rare, right? You cannot get that genetic power. But with an IVD directive, you are forced to validate to the fullest of your capabilities that diagnostic test. And if there is a false diagnosis that's reported in the future, it is the manufacturer's responsibility. So what you have in the UK at the moment for most tests that are taking part in genetic medicine departments, not so much virology, they're different. But in genetic medicine, they are self-validating their own tests. And under the new IVD directive in the EU, that means they are the manufacturer and they hold the risk. And this is something that's a very uncertain picture in the UK. Like, what are the UK government going to do? Are they going to adopt IVD directive? And how's that going to impact the tests that are currently offered? Because none of them are currently at the standard that would be required.
But
It's
actually,
an unanswered question right now.
how does it work the other way around? Say, if there is a German manufacturer would like to bring a test, or vice versa, a PIS assay, I think you are working quite international, isn't it? Having sites elsewhere. If you develop a test here, just across the road, do something, say, in the US or elsewhere where you have the presence, or just outsource a test and somebody adopts it, who then?
Yeah, so at the moment we are working towards the IVDD standards. However, for the UK market, I think we're looking at UKRA,
Right.
UKCA. So yeah, those are the directives we work under. But yeah, it's certainly a challenge for us. It's something we're figuring out at the moment.
That sounds good. Thanks for the comment, Sam.
I got a question regarding patient data. Since data is essential for companies, shouldn't patients have the right to actually sell and profit to their own data rather than, yeah.
Not quite sure how to answer that.
Sorry, that's exprompt.
The profit to like our commercial profit, or say if there is a joint venture in the future between our company and a pharma company, and we identify a biomarker that kind of leads to drug development. Is that what you're saying?
I assume smaller biotechs do buy the data from some other companies that provide the service, but the patient doesn't see any of this profit from their own health data.
So as a strictly commercial question, we provide a service to the patient, and that's what they pay for, and they get that service. hopefully they're happy. Some patients are not happy to relinquish their DNA data, and that's absolutely fine. So we only use their data under consent. But that is probably an area I would say that's very underregulated. I don't think profit has ever come into it. It's usage rather than profit. You could take that argument further and say, is DNA something that if a company holds it, and lots of companies do, not just genomics companies, is that a commodity? And should the patient benefit from it? I don't have the answer to that.
Thank you.
That's all right.
Any more
That's a good
takers?
question.
I was just going to
Any more
say, we
takers?
don't let patients sell their organs for a reason. I don't know if we would consider your gene data as something as valuable as a kidney. And how do we regulate that? I think, again, it needs more oversight.
Data is the new oil, right? That's
Yeah.
why all this data is connected. Like Google knows you're here now.
And I feel like there are
So-
plenty of people that pay to give up their genomic data, right? With 23andMe and Ancestry. And what kind of wealth of data that these companies have that is locked away from research institutions and small biotechs, that like people have, paid £123 to do it, and they're not getting any profit from that. So I don't know, I think it's a big ethical question.
But
No, you have my.
no, it's a separate point.
Yeah, again, taking that argument a bit further, it's, biopsies have been taken from patients, for as long as kind of the NHS has existed. And if we use data as kind of currency, that would open the can of worms for biopsies and other things that potentially belong to patients. Wow, yeah. Interesting.
So
Scary.
in terms of the sort of data side, if I make a model, like a machine learning model, classifier or something, that depends on all the data I feed it. If I build a model and then suddenly I need to remove that sample because there's an issue, you have to regenerate the whole thing, and that it goes through the sort of whole part of the research process in terms of papers. You remove one sample out of 100 and suddenly your differential expression will change, and therefore all your numbers will change because all your p-values change because of the false discovery rate, and all this kind of could change everything. And so if you can go, well, actually, I did share my data, but no, I don't want to, then that causes big problems for that as well. But you can retract data from repositories. There are certainly some ways of doing that. But yeah, it's a issue for the data side as well.
I just wanted to add as well, I think it was Sir Patrick Ballant at the Festival of Genomics talked about the report just released by the government on this issue, and also thinking about whether, for example, insurance companies would then be able to use this genetic data to define your premiums, for example, or whether certain predispositions to neurological disorders could affect the way the law sees and interacts with you. So it's certainly questions that are being thought about by policymakers, and the very big questions. And yeah, there's clearly no easy answer.
I think where the scariest, I personally find it, it's, sometimes even in sort of not marketing world, but when you subscribe to one, say, newsletter, or you bought something from the shop X, and then they say, hey, but now you're subscribed to 20 other partners, highly selected partners, but you have no clue who are these highly selected partners. And then one day I understand that I need to delete like most of my emails per day because it's all kind of junk. And I was like, I have no clue where they're coming from. So I still don't know where these highly selected or who is the one who selected them so highly. But unfortunately, they all ended up in the bin for me. So that's where it's kind of can be scary, because if I don't know, I don't want to say 23andMe, but if a genomic company partners with insurance company, it is quite scary, because as soon as they know all these biomarkers and can track back to these people, suddenly people can see their bills like increasing tenfold.
Actually, Agustin brought up the word insurance. So what if your genetic profile could actually reduce your premium?
I'm not saying it should be used one way or the other. My point was that it is being thought about, and policy will be involved in making these decisions. Yeah, it's difficult. I think ultimately it will come from patient consent. So if you consent to your disclosing your data to insurance companies, then maybe it can be used. But again, that's my personal view, and I don't know enough about the topic.
I would say you can't have it both ways. You can either say it can't be used at all, or it can go up and down. And if you're buying life insurance and it's suddenly twice the price, you probably don't buy it. So that's a bit.
Sorry, if I may add, if this data is so widely used now that even insurance company have access to it, what if, for example, like, you know, we start categorizing people depending on their genetic profile? Like, for example, athletes, you just categorize a 15-year-old who is dreaming of becoming a professional athlete based on their genetic data. And sometimes this genetic data could be wrong. If this kid was given the opportunity to actually train and become a professional athlete in the next 10 years, he could actually become a very good athlete, maybe one of the top athletes in the world. And we can't just rely on data to determine this.
Yeah, but I think then, sorry, I don't want to challenge you, but then the question is, if it is extremely promising baby is extremely lazy and all they do is watch the Netflix, right? And the parents say, oh, I was told you will be Olympic champion, but all you do is sat here. You don't even go to, I don't know, wherever, to the gym.
I think it opens up huge cans of worms, full stop. And you start going, well, you can't have this job because you're disposition, like you have this gene that suggests you may have a more increased chance of certain trait.
I think
like,
there's one
racial
huge
profiles.
aspect of this we haven't really talked about yet, which is if we're building all of these AI machines to make decisions based on our genetic data, what's the genetic data we're feeding in? Because at the moment, there's a massive sequencing gap. Like almost all of the sequencing we've ever done is on people of European descent. And so how can we possibly start making these decisions and implementing these algorithms if we don't have a like rounded data set? I mean, currently we're sitting on a very biased data set, and we know what biased data sets do to AI. We've seen it in the tech industry all over the place. And I mean, I've been really fortunate to have discussions with this really amazing researcher here in Manchester called David Wedge. He's kind of helping to close that sequencing gap in cancer genomics, where he's kind of found that actually patients of South African and East African descent have wildly different mutational profiles and would require completely different treatment programs than what people of European descent need. And so if we're going to extrapolate all this genomics data into making like small life decisions for us, I think we really have to address that gap before we do anything.
I think you're absolutely right, Kath. I mean, and we as a company absolutely acknowledge that. And we sponsored a number of programs to undertake population screening in a number of countries, because even in Europe, there are distinct populations where you cannot extrapolate from a Caucasian population, Israel being a good example. So I think there's within the industry, our kind of industry that's building some of these AIs, there's a recognition of that and a drive towards capturing more population scale genomics, which is why it's so important to democratize sequencing, right? I mean, if a genome was to cost still 3.9 million, there's no way on earth you would do that. But at $100 a genome is far more accessible. And I think you're going to see much, much better tools emerging in the next 1 to 3 years as a result of that data that's coming in now.
Interesting.
Yeah, so the having an appropriate control set, like, so I'm building classifiers for subtyping cancer from liquid biopsies, and our controls are collected from Manchester as like risk matched, but then it wouldn't necessarily apply to somewhere else. Like it might call them incorrectly because it's not got the correct ethnic background from that it's seen in the fitting process.
My actual site here, it's not my quote, but NIHR quote, again, based in City Labs. And they say they're very privileged to be based here in Manchester, because if somebody wanted to organize a clinical trial, they could not go anywhere but do it here, because here in Greater Manchester only, there are 200 languages being spoken. So it is a fantastic library, if that's the right way to say, and hopefully kind of in a good and bad way, maybe it supports you getting access to samples of people having a variety of backgrounds rather than just.
The trick is getting them to consent to and collecting from a variety of
And not
backgrounds.
losing a sample.
I would say as well, part of the issue is the quality and how complete the metadata associated to this is. So if you don't have very informed metadata, you might lead to a bias in the machine learning algorithm that are actually non-causal. But if you're not collecting data on socioeconomic status, profession, or less visible phenotypes, then you might be missing stuff out. And the problem is that the metadata then is not updated either. So
I mean, metadata is a huge problem in itself. Like you get data samples and you're missing huge chunks of the metadata around it, and it's not conducive to good fitting.
Yeah, I totally agree. Actually, clinics have different ways to collect metadata for patients, so it's very difficult to get a hold of the same type of metadata from data that derive from different areas of the world. Yeah, it's just there is no clear practice that is applicable everywhere.
I wanted to ask, oh, sorry. No.
Yes, I think that the trend here from what we've been saying is there need to be more guidelines in all of these areas. And yeah, metadata is definitely one of those areas where we collect what we think will be useful, but what will be useful in 5 years might be something that we haven't collected today. Again, it's a completely unregulated area.
But actually thinking outside of the box based on the example of Bitcoin, I didn't meet these people, but I read crazy stories, right? When there were these guys who almost knew about Bitcoin in like 1990s or whatever and invested, and now they live somewhere, oh, they are somewhere nice. Does it mean that actually maybe absence of regulation helps to do, obviously it protects, but does it not help as well maybe to discover more things, because you have almost all the options in the world, you do what you want to do, but I think then it comes to the ethical question and boundary, right?
I think, it's safe to say within this company, yes, we have the flexibility to do more, and we probably wouldn't use that data in an unethical way, but without regulation, I think that's the danger that you have, that, in the same way with, doing work on embryos on blastomy, and that's very tightly regulated.
Mm-hmm.
We don't want
To breach.
We don't want breaches of kind of going beyond what is ethically acceptable because there are no regulations there.
No, of course, that completely makes sense. You don't want to go that way.
And I'm not sure that Bitcoin and, for instance, like social media is a great example of,
No.
of the what happens when you don't have regulation in the technology space.
No, absolutely. I wanted to ask I wanted to discuss actually bottlenecks, but I think we had a very provoking conversation already, and we heard what the keynote speakers said. But in a nutshell, so we discussed the challenges. Can we collaborate? Like, can we work together? Can we bring more partners? Can we expand our network so that we can solve these problems? Is there something we can do? Literally go home, wake up tomorrow and like, oh yes, I could approach this and that person so that we can start, working together. Maybe they can advise with other steps that I can't find answers to. Is there a way, between collaboration between academia, private companies, investors, policymakers?
I'd say part of it is standardization. So the data collected and the analysis done can be reproduced in other places, and the data can be used in the same fashion everywhere and hopefully be future-proofed. Yeah.
So that means we need to speak more. So every one of us collects the same data.
And well, not the same, but follow the same procedures.
Sorry, same procedures and protocols, or these kind of work within the same sort of frame.
Yeah, to a certain extent. And I would also say work more with the key stakeholders for, in the end, the clinical setting, so that we're not making products and in the end don't get adopted into a clinical setting.
Yeah, I was going to say that as well, about kind of from the very start of a research career, know what the clinical need is. I think there's kind of a gap there where we don't really think about it all that much, especially from my own experience. And then I think in regards to what our first speaker was saying about the Pankhurst Institute being a site of multidisciplinary teams, it's not always easy to build them, especially in academia. I think it's very hard to attract computer science graduates to academia unless they're like desperately wanting to do a PhD because the money's not there. And so actually that's kind of important, where you could build collaborations between academic labs and tech companies, where kind of you get the talent from both sides and both are being compensated and both in an industry that they kind of want to progress in. But then you still get to build a multidisciplinary team. And that might be a better way forward, maybe in building those kind of groups.
Very interesting. Any more? No? Any questions from the floor or from the Zoom?
It might not be very related, but with the development of artificial intelligence and its ability to classify whether a patient will be prone to cancer, for example, or not. Do you think it would be possible to kind of circumvent doctors, and that artificial intelligence model will actually make the prediction and even predict the drug that would be suitable to the patient?
But then who will be responsible for it? That's my actually next question to that. Really good question.
I think we're quite a long way off that. And I don't think that some human interaction is going to go away anytime soon. So just because the computer says something doesn't make it true. And for instance, a prediction for a female patient as having prostate cancer is one that comes to mind for me recently.
Just a comment about what you said about future-proofing, just everything to do with analysis. And I guess, is there anything being put in place for that? And just I wanted to elaborate a little bit on that. And in what ways can you counteract that, like in the future, be it the data set being removed or retracted or anything like that, but also just within the analysis itself that allows you to, or even how you capture all the sequencing and everything that allows you to future-proof it in the future. So is there any comments about that at all?
think it's hard enough to current proof it, to be honest.
Like we lose data sets within 3 years in our lab. Like, how are we supposed to future-proof access for decades in the future? It's just madness. I mean, the data structure just isn't there, I think. Yeah, especially within research institutes as massive as the University of Manchester. You've got a lot of data being generated. We really need like investment into our IT and computer science core facilities for that kind of work to be done and for expertise to be in those fields.
That means we need more investment.
Yeah, more money.
It always goes back to the same kind of
Fortunately,
square one. Money, everybody needs those.
But fortunately, DNA is quite a durable matter, so we can always resequence. I think probably, you know, Catherine and Simon are right. We'll look at some of the data sets that we're doing today in a few years' time and say, you know, can't believe that we accepted that, you know, that passed the quality. But
how did we get anything out of that?
Any more questions or Zoom?
Actually, I want to raise something. It's a general comment, but that is something that we are kind of addressing at the same time as we're performing our analysis. Is how, I mean, we are, I would say, a selective group in that we understand genomics and, you know, there's no kind of jargon for us, but how ready is the British public, I would say, or any public really, to accept this as part of a new reality? So some of the scenarios that you brought up, circumventing doctors or sequencing a baby at birth, how ready do you think is the public to accept this? I don't have an answer for it. I actually want your opinion.
Because we already have some example of this. For example, during COVID in the NBA, they implemented the wearing of rings that could possibly it says was whether there. So everybody was scared, or the players were scared, whether these rings could actually get more data on these players' physical data, maybe injuries, genetic data, any kind of data. And this data could be shared, and these athletes would be scared for their contracts. For example, if they could find out that they are injury prone, someone wouldn't want to sign them anymore. And these guys, their entire life is based on this career at this point. So if this caused such a panic in such a small group of like 20 NBA teams, just imagine a population of people from different backgrounds. So yeah, it will be crazy, I guess.
I was actually thinking just to maybe try to answer your question, or just thinking out loud, you know, do you think having more voice in the policy from sort of scientific community will help? Because the reason I'm raising that, about half a year or so ago, I think NHS or the government basically said that now everybody automatically is registered as a donor, because they knew there is a mass lack of donors. So now automatically everybody is enrolled unless people tick the box that they don't want to be. Do you think maybe one day when so many scientists or, you know, academics and clinicians will say, you know what, we need that data, please sequence every single baby that is born, you know, maybe they will say, sorry, but your baby is sequenced because by default, unless you tick the box now.
Yeah, you will be sequenced unless you opt out.
Yeah, because it will be so cheap, thanks to Sam.
Not me personally, but I mean, newborn screening already goes ahead, right? That's what the heel prick is for. It's just that it could do so much more for a really, really good health benefit, right? I wanted to mention though that one of the gifts, the many gifts that COVID has given us, if you can put it that way, is that now words like variant, genetics, host genetics is now part of popular culture. People get it.
PCR.
Yeah,
Oh,
look,
yes.
everybody knows, everybody's an expert on PCR now. And I'm an optimist, I know this, but I think that's incredibly powerful. I think we are going to see the pendulum swing a little bit in people that are in favor of, you know, if I can get a genetic test done that tells me which drugs are going to work and which dose I should go on for my blood thinner, example, then, and how many paracetamol I can take before it kills me. I mean, why wouldn't you want to know that? That's so powerful, but the ethics behind that, the policy behind that, oh God, it's got a long way to go. And never mind the electronic health records. They're just not robust enough to capture that information yet. But it'll come, I think.
It sounds like it's, if you get the marketing message right,
Yes.
we can convince people that actually it's a good thing to have your
Well,
genome sequence.
People opt in for 23andMe. And that's to see whether you'll be bald in 20 years. It's not life-threatening.
I'm not saying about dogs, you know, when I was a child, I remember walking the dog and there was like a random dog passing by. You try to ask what's this kind of breed and they say, oh, I don't know, it's just like whatever. And now I asked one day somebody and then they almost told me percentage of the makeup of that dog. I was like, excuse me, you know, I understood what they said, of course, but I didn't expect that to hear that answer exactly in the park. I was like, okay, that's good to know. Sorry.
That's Microarrays as well.
Any last comments before we close?
But the other part of what has been clear through the pandemic is the people who don't want new things like this and who would be very vehemently opposed to sequencing everybody and the big data being in some faceless location. There were already conspiracy theories about everything.
But what I like the most, actually, I completely agree. Somebody said that marketing did the job and I was amazed. I remember I came to the office. Most of the people are not having a background in biology or chemistry. They're like surveyors. And I overheard the conversation of my colleague who the other day was asking what's the difference between GMP, GLP, and all others. Like, okay. And then suddenly I can hear almost professor-like intonation explaining to his other colleague who is sort of similar level how PCR works. I was like, oh, okay. And then it was really interesting to hear, you know, so yeah. Well, I wanted to say thank you so much for my lovely panel discussion. It was really thought-provoking. And actually, thanks a lot to attendees who decided to stay. And I think it was really good to interact as well. So thank you.